I'm going to talk about epithelial cell dynamics. But first, what are epithelial cells? The epithelial tissue is a type of tissue that is made of a thin layer of cells that cover and protect organs. You can see epithelial cells every day. It's your skin, for example. Although an epithelium can be made up of several layers of cells, we'll only be looking at single layer epithelium. When you grow or get a wound, the epithelial cells multiply and rearrange themselves. Let's put some cells in a petri dish. At first, these cells do not fill the entire dish and they multiply. As there is still a lot of space, the cells can move by themselves, individually. Then, when they fill the entire dish, the cells start to move together, and that phase is what is most interesting to us. At some later stage, they jam and the movement almost stops. Let's look at that second phase we talked about. When put in a square dish, you will see that each cell starts to move in an elliptical pattern. But the most interesting part is that cells move in parallel trajectories, kind of like people waltzing in a ballroom. When the cells move closer to the edges, their density increases and the sheet gets thicker. Another study shows that the denser the cells, the slower they move, and vice versa. Or also, if you introduce some sort of external strain, compressive or extensile, the speed of the cells also decreases. A very curious point emerges. Individual cell motion is non-inertial, yet the behavior we saw reminds us very much of inertial systems that we are used to, such as fluids or crowds. The study of the dynamics of the epithelial sheets is essential in the understanding of the mechanisms of morphogenesis, wound repair, but also cancer.